All right, thanks for coming. My French is not good enough to even follow the presentation of Professor Chazelle earlier. But he told me that the theme is to study algorithms with algorithms, and I continue the theme here, introducing a piece of mathematics which you can think of as algorithm, and studying some natural processes. So this is about persistent homology applied. As Bernard said, I'm from IST Austria, currently still on leave from Duke and Geomagic. And this is work that accumulated over the years, and the main contributions are collaborations with the early work with Cecil Delfinado and Ernst Mücke, and then the mathematical work with David Cohen-Steiner, John Hera, and Dmitry Morozov. And the last part of my talk will be about a biological application, and this is work with Carl Philipp Heisenberg, Michael Kerber, and Gabriel Krenz. And there'll be three parts to this talk. The first is kind of to get you into the mood. The second one is going to be mathematical, and the third one is going to be biological. And the mathematics is motivated by, I guess, looking at nature or thinking about nature. So I'm kind of convinced that the concept itself, when you see it the first time, it probably seems strange to you, but I'm very convinced that it's inside you. Somehow it's very natural if you see it the right way. It might even be encoded in your brain. So in general, mathematics, I feel, is this way that the concepts that are useful are very natural, but they are encoded in a way that we don't think they are, but they still are. So I hope in the motivation part to give you a feeling of what I want to do in the mathematical part. So here's just some discrete set of points, and it looks a little bit like a circle. And to work this out, I'm going to grow the points into little disks. You see, as I grow now into little disks, nothing much changes. But if I grow them further, this is almost, it's already connected. You see a few holes here and there. Maybe I'll point them out. So you see a big cycle with surrounding a hole, and then some little cycles surrounding a hole. And in natural circumstances, when you see a natural object, you often want to say there's one hole instead of 8, maybe. And indeed, if I grow the disk a little bit further, the little ones disappear. Only one remains. But this does not mean that if I grow further, it stays this way. So if I grow a little bit more, there's a second hole appearing again. But not for long. And then, if you keep growing, nothing happens. So if I do this fast, you see really just one cycle. And I wanna capture this mathematically in a general way, and also so it's applicable for many situations. And one situation I want to talk about. And I'll tell you a little bit more about where the idea comes from. It was some 20 years ago. We looked at proteins a lot. So here's a protein. This is this is hemoglobin, transports oxygen. So usually people model them as little spheres. So you can take the picture from earlier and do it in 3 dimensions. In particular, what I would like you to think about is, if you have these spheres, I would like to turn it into a simplicial complex by the nerve operation. So this is a union of balls, union of spherical balls. And I'm going to cut it with the faces of the Voronoi diagram of the points. Perhaps you can imagine it. And this cuts this hemoglobin into convex pieces. Namely, each convex piece is a sphere or a spherical ball cut with half spaces. And then if they're neighbors, I would draw an edge. Or if 3 are joining along an edge, I draw a triangle. Or if 4 meet at a point, I draw a tetrahedron. And to illustrate this in 2 dimensions, it's a little easier to draw in 2 dimensions. I have the same set of disks. I draw the Voronoi edges as to the extent they are visible within the disks. And then I draw the dual. Okay? So now you see nicely the big cycle and the little cycles that we don't really want. And now, if you remember, we grow the disks 1 step. The regions that are connected get more because they get larger. You have more connections, so a few triangles, they all fill up the holes. So this transformation, taking the nerve, is very useful, and I'm going to use it in particular when you have convex sets, because it maintains the topological connectivity of the object, more precisely the homotopy type. And since we talked to biologists, in particular biochemists at the time, we thought we can turn all these algorithms into practice and then find the big features of proteins by computing homology. So we learned homology, and then we developed an algorithm. Here's the algorithm. It's an incremental algorithm. It's very fast. It's very simple also. Namely, I add 1 simplex at a time. In 3 dimensions, there are only 4 types of simplices. Vertices, which are points, edges, triangles, and tetrahedra. And if I add a vertex, I get a new component, which means beta 0, which is the 0th Betti number, or the rank of the 0th homology group. If you like, it's also the number of components. So that goes up by 1 if I add a vertex. If I add an edge, now there's 2 possibilities. Namely, I could connect 2 components. Beta 0 goes down. Or I connect 2 vertices which have already been connected somehow in the complex, and then I create a cycle. Beta 1 goes up. So Beta 1 counts the number of tunnels in 3 dimensions. And triangles behave the same way. Namely, a triangle could either close a tunnel, then Beta 1 goes down, or it could complete a closed surface of some type, like the last triangle of a sphere, for example. Then Beta 2 goes up. So Beta 2 counts the number of voids in this shape or complex. And the last thing, when you add a tetrahedron, you fill a void, and so Beta 2 goes down. It's a very simple algorithm. All you need to do is this case analysis, binary case. This generalizes to higher dimensions, but has trouble in implementation. But in 3 dimensions, it's fast. So we could take proteins, feed them into the computer, and figure out the homology, the connectivity. And then we ran into the following phenomenon. So this is grammacidin A. It's a protein, a very small protein. It's a cell membrane protein. You see it in the shape. So this is 6 times the same protein. It's only left and right is 2 different angles of views. And from bottom to top, I increase the radius before taking the nerve. What you see is the nerve. But what you see is there's this tunnel that goes through here. Here you see the tunnel from the side. And this tunnel is embedded in the cell membrane. And then there's ions channeled in and out. And then somehow you would like to say there's 1 tunnel, the rest is not important. And I wanted to have, so this was 15 years ago, I wanted to have an algorithm that detects that. But then what does the algorithm do? It gives me all kinds of measurements. So this, let's see, this is for grammacidin a record of the number of tunnels. So from left to right in this picture, I grow the radius of the spheres. vertically, I count the number of tunnels. I think actually the logarithm, if I remember right, the logarithm of the number of tunnels. And in this marked field over there, you see that at this point, the 530th complex has 34 tunnels. And most of the time, you have a lot of tunnels, so around 30 to 40, plus and minus. I wanted to see one tunnel, but I saw 30 or 40 all the time. And that was kind of the trigger that there must be some logic to filter out this one important tunnel from this mess. And some years later, we had this idea of defining persistence, which I will do in the second part. And then we could draw this diagram. So this diagram is the same as what you just saw, only one extra dimension. Namely, from left to right, again, I grow the radius. Vertically, I count the number of tunnels, again, taking the logarithm. But from back to front, I increase persistence. And in a nutshell, what persistence is that for each tunnel, it keeps track of when it gets created and when it gets destroyed. And the persistence of it is then the time lag between birth and death. And you see a big triangle coming forward. This is the one tunnel that determines the function of grammacidin A. That was the protein. All the rest is, I call it topological noise. Some lives a little longer, some lives a little shorter, but it does not live as long as the features. This is what I want to explain. And then for biology, the application, I go to 4 dimensions, and then that will be something completely different. All right. I did notice that during Bernard's talk, nobody asked a single question. So I don't mind if I ask a question. So let's talk about persistent homology. Introduction by pictures. I guess I can't really assume that you know homology theory. That's part of algebraic topology, but I want to do it such that everything is elementary and you can follow without the background. My example will be, this is a torus, a surface, and just to make it a little bit more interesting, I added a nose on the right. So I call it the torus, the upright torus with a nose. And the Betty numbers of the upright torus with the nose are, first, it's connected. So Betty 0 is 1. It has 2 tunnels. One is the one that you can reach through. The other one is inside. You can reach around the torus inside. That's 2. And then Betty 2 is also 1 because you have a complete surface without boundary. Okay. I'm also going to look at what we call sub-level sets. So I'm taking a threshold. I take the part of the torus below the threshold. I look at the Betty numbers. So in this case, we have 2 components. Betty 0 is 2. I have only one tunnel, namely the left side is a cylinder. The rest, the right side is just a disk. And Betty 2 is 0. No closed surface without boundary. I'm also going to take homology or Betty numbers of the boundary of sublevel sets. So these are the 3 circles. So there I have 3 components and 3 tunnels, 3 and 3. I'm also going to take relative homology. So this might be a little bit harder to introduce first. Relative homology is you have a space, the sublevel set, but then it's open at the boundary. So if a curve goes up to the boundary, you actually think it's a circle or a loop. So you count it as 1 for Betty 1. So you get this one loop on the left, because you can go up and then the boundary disappears because it's open. And there's two closed surfaces, because both of them, the boundary disappears. If you're not happy with that explanation, I can understand that. But I want to point out that this vector 0, 1, 2 is the upside down of this vector 2, 1, 0. And indeed, there is a theorem called Lefschetz's duality, which says that these two vectors are transpose of each other. So if you're not happy with relative homology, you can go back to the homology of the sublevel set and transpose the vector, you get the same numbers. But there's something called excision, which is very natural, going to be useful, which says that if instead of the pair y, y0, I take the whole torus instead of y, and I remove from it everything above the boundary of y, I get the same result. Because I really only see the sublevel set, which is blue, and what happens above, I don't really know, and I don't really care. But there's a formal theorem when this is true called excision, and I'm going to use that. Okay, so this was a brief introduction into homology groups. Now we're going to use this with pictures again to define ordinary persistence. And maybe we go through this exercise and then I try to generalize it a little bit so that you have the right idea in your mind when you see this. So in ordinary persistence, I'm going to look at a threshold, and I take the sublevel set below, and I look at the Betti numbers. And then I'm going to increase the threshold all the way up. At the moment, it's down here. I don't see anything. All the Betti numbers are 0. So from left to right, you have 0, 0, 0 for Betti 0, Betti 1, Betti 2. Now we have a disk, so one component. When we touch the nose, we have a second component, 2, 0, 0. And then we hit the lower saddle of the hole, at which point one cycle got created. So on the right, I keep track of the cycles that are counted by the Betti numbers. You see where they are born, and they keep on living, going up. I was told that when people yawn, it's actually because they're thinking very hard and they need oxygen. So I'll take the positive interpretation of it. Let's take one more step. We hit the saddle that separates the nose from the rest. Here, the two components merge. So something interesting happened here. Namely, one of the components died. And by definition, always the younger dies. So in this game, being young is not good, because you always lose out against the old. And at the upper saddle, another one cycle is born, and at the top, a two cycle is born. So this is the homology of the torus. If you look at the vectors that go to infinity, 1, 2, 1, and there's one class that got born early, but then died early. This is the nose. Okay, so this is what we call ordinary persistence, but there's more structure to be discovered, which you'll appreciate later. Namely, I would like to follow up with a second pass so that everything that gets born also dies. We don't like things to survive forever, because if something survives forever, then you don't know how to take the difference between death minus birth. We'd like to have a number. We don't like infinity. Okay? So we follow up with the second pass, which goes backwards with relative homology. And first thing, the component dies. Second, one cycle dies. Third, a two cycle gets born. So it's possible that going back, something gets born. The reason it's born is because the component of the sublevel set separates out. The boundary disappears by definition of relative homology. So I could actually have two complete surfaces in my hands. the second 1 cycle dies, the 2 cycle dies, the other 2 cycle dies. Okay, so after we come back to where we started from, the Betty numbers are again 0, 0, 0. Has to be this way because I removed everything. I'm again talking about the empty set. So this puts me in a fortunate situation where everything that is born also dies, and then we can measure differences. Now, I left a few more abstract slides in here because the important thing about this exercise is this is extremely general. It's not only for height functions, it can be done for any function. For example, at the introduction, I showed you some proteins, and I talked about how to grow the atoms, so then the shape of the protein changes depending on the radius that you take. In this case, the function was the distance from the centers of the atoms, because a sublevel set is exactly a spherical ball about around each atom, and the radius is equal to the threshold. So you can imagine this maybe by imagining in 4 dimensions. You have your protein sitting in 3 dimensions. You have vertically the distance function, and then you do the sublevel set. But really, any function you like can be done, and you get some readout. Okay. This whole thing leads to an algebraic setup, which I now explain. So here's a function. X is some topological space, maybe R2, maybe R3, some real numbers on it. We look at sublevel sets, which I call X sub I. And the double bars are the spaces. And the X sub I on the right, which doesn't have a double bar, is the homology group. And if I use field coefficients, then this homology group is a vector field. A vector space. So I'm playing a lot with vector spaces. For completeness, I need superlevel sets. So this is when I come back. And again, I take the vector space, which is taking the relative homology of the pair, X mod out the superlevel set. And algebraically, this looks like this. From left to right, you have the vector spaces. Each vector space represents how the topological space that you look at that moment is connected. And the vector space is just a normal vector space with some generators. Each generator is a cycle, and there's nothing magical about it. The one interesting thing is we have maps from one vector space to the next one. They come from inclusion, because my spaces, the way I create them, always get larger. So every cycle in a space exists in the next one. Could be trivial, but maybe not. So we have this forward map. And we use this map now to have a formal definition of what I introduced earlier, informally. Namely, when I take the image of xi minus 1 in xi, and I find a class alpha which is not in the image, then I say alpha is born at xi. Okay, so this is when we see a new class in the filtration. And then we can follow forward for a while. Alpha will stay, or the image of alpha will stay outside of the image of xi minus 1 until it eventually gives in and ends up in the image of xi minus 1. Then we say alpha dies entering xj. And eventually this happens, because this list of vector spaces starts with the empty, or with the 0 vector space, and ends with a 0 vector space. So there's no way to escape. Okay, so this is the formal definition of something is born and something dies. And the important thing to remember from this is everything gets paired, and there's always pairs, and they're canonically defined. And the meaning depends on the meaning of the function. I need to introduce something called the persistence diagram. It's a way to keep a record of what we're doing. Kind of the bookkeeping. So here's our exercise. the intervals going up and down. The way I'm going to draw the diagram is I draw the intervals from left to right, and I unfold them, make them straight. And then I put a big triangle on top, right angle triangle, and then I replace each interval by a point. The point is the top point of a right angle triangle that goes down to the interval. So I replace the 6 intervals by 6 points. The numbers that are marking the points are the dimensions of the cycles. So 0 is for the components, 1 is for the one for the tunnels, and 2 is for the voids. And then I remove the triangles and the points remain. So this is what we call the persistence diagram. One thing I like about these points is you see the symmetry nicely. This point set is symmetric relative to the middle axis. From the intervals, it's not so obvious. This is a consequence of Lefschetz duality that I mentioned earlier. When you do the reflection, though, the dimension changes. So 0 becomes 2. 1 stays 1 because the dimension of the space is 2. So this is true for 2 manifolds. I should have said that. If you feed something to this algorithm which is not a 2 manifold, then you don't have symmetry. There are some more complicated symmetries, but I'm not talking about them. And since there are 2 manifolds, so the complementary dimension means 2 minus the dimension. All right. I've drawn this triangle in a maybe peculiar way, namely I decomposed it already in 4 triangles, which I call ordinary. Ordinary was whatever was born and dies going up. Relative is whatever is born and dies coming down. And then there's stuff that gets born going up and dies coming down. This lives in what we call horizontal and vertical. Why I call it horizontal and vertical, I'll explain a little bit later. But these distinctions have a big significance for the application that I have in mind. So I introduced it here. So I call this diagram the persistence diagram, DGM of X, where X is this tower of vector spaces. And for the mathematicians in the audience, it's maybe satisfying to know that if you have 2 towers, X and Y, that are isomorphic, then you get indeed the same diagram, if and only if. So these diagrams characterize towers of vector spaces. It's actually very easy to prove, but it's good to know. Yeah, so before I get to the biology, I need to tell you a little bit of how to read this diagram. The diagram encodes the Bethe numbers of all kinds of spaces. So of course, the sublevel sets. So here I marked the sublevel set. You can read it by drawing a rectangle with an anchor in the ordinary diagram. All the way this way, that way, it's a rectangle. You count the points in this rectangle. What do we have? A 0 and a 1 in there, which means that there's 1 component and one cycle. No surprise, it also counts the Bethe numbers of the relative homology group, relative to the superlevel set, by a rectangle that just sits on the other side of the diagram. Here I have a 1 and a 2, and indeed the Bethe 1 and Bethe 2 are both 1. A little bit more difficult is to see how it counts the Bethe numbers of the level sets. For that, I take the symmetric difference of the 2 rectangles, and so I get 0, 1, 1, 2, which doesn't quite correspond to the 2 components in the 2 loops, except it does, with one extra twist in how you count, namely the vertical diagram, you only see slices of cycles. And I find this actually interesting. So this has all kinds of consequences. So let me talk a little bit about it. In the torus, there's 2 loops or 1 cycles. One goes around an arm, and the other one goes around a hole. The one around the arm you can see in a slice as we move the threshold up in a level set. The one around the hole you never see, because in a slice. All you see in a slice is that you have this loop and you slice it, so you see 2 points perhaps. So this one-dimensional loop you see as a component or as a generator in homology 0. So the vertical cycles, they all demote the dimension by 1. So then I read 0, 1, 0, 1. And this is exactly the Betti numbers of the level set. And the reason why I mentioned this is because I wanna do the biology in space-time. And time is gonna be the function. And so a level set is then the object at a certain moment in time. And this is how we see the world. And so if I take the world or people or animal or cells and then map it into a 4-dimensional space by adding time to it, so it's a solid space in 4 dimensions, and measure homology this way, then I can see 2 types of cycles, namely the ones that we see at any moment. And this is when you observe the world, you see it happening. But then there's the connections you don't see in the world. Unless you think, namely things that were connected, disappear, connect again, disappear, connect again. For example, relationships are a little bit like this. You see 2 people, they talk to each other, nobody thinks anything about it. In an hour, they do it again. Then you think about it, next day again, you think there's a relationship. This is a vertical cycle. All right, so this was the end of mathematics for today. No questions, all right. Let me talk about applications. So there's tons of applications of persistent homology, but I didn't want to bore you with too many. I just want to talk about one, and this is about cell sorting. And cell sorting, I brought a little movie to show it, but actually I didn't have to because Bernard had a lot of spatial sorting algorithms running on his movie. He had the fish swarm going around, points moving around. So the question I would like to ask, and this is what this method is about, when something moves around, I would like to know what happened as they moved. What patterns can I see there? Such as if I see the same fish moving again, how can I tell whether the fish now move different? Because maybe they are afraid. Maybe there's a shark somewhere. Or somehow the program changed. Or whether it's the same. And specifically in this example, I want to talk about cells. So these are cells in the development of a zebrafish. There's 2 types of cells. At the beginning, those cells are kind of mixed together, and then they move around, and magically, after a while, they separate. And then they develop their own organs. And so the question is, how does this happen? Why do they separate? But also, if I change the genes a little bit, and so the cell sorting mechanism is impacted, I would like to be able to measure how it's impacted and tell the difference, if there is one. Okay, so here are 2 cells. They have a nucleus. The biologists I work with, they make them fluorescent with different colors. So they have a palette of colors. I'm just going to use 2 colors, red and green. For some people in the audience, red and green might be the same. So I think later I use red and blue or green and blue. Just sometimes I'm not sure myself. Okay, so then they stain the cells, and then they take microscope images. So those are a time series of 3-dimensional images. Okay, so the space is 3-dimensional, and then we have a 1-dimensional time, so it's going to lead to 4 dimensions. So I model the whole thing 4-dimensionally. And I want to say a little bit of how we do this. So here is our 2 collections of cells. I only draw the nuclei. And from the because that's the only thing we observe, and we know from which type they are. A very crude modeling is taking the Voronoi diagram. So it's a bichromatic Voronoi diagram. Each nucleus has the region of nearest points, and I call this the cell. Actually, what we do is we also draw a radius, so I clip the Voronoi cell to within a certain radius, and then that's what we call the cell. But I want to ignore this for now, just to simplify things. So I have my bichromatic Voronoi diagram, and I draw my bichromatic Delaunay triangulation. So this happens by connecting two points in the Voronoi diagram if they have the same color and share an edge. Triangle, if I have three of them, sharing a point, having the same color. Okay, so I have a green and a blue, I think. And those things move. So then, also, you should imagine this in 3 dimensions. So we implemented what we called a kinetic algorithm. So this is a standard algorithm technique in computational geometry. But we had to apply it to these complexes in 3 dimensions. So I'll show you what it means. So suppose only one point moves. So I don't want to have everybody move because it's too confusing. So only this one point up there moves. If it moves, see what happens here. This edge shrinks down to 0 and becomes the orthogonal edge. When that happens, these two cells get connected. So I should draw this blue edge and also the triangle. Okay, let's move the point a little bit further. And before I change the complex, let's see what happened. In this case, this edge shrank down to 0 and gets replaced by the opposite edge. So now I have this region connected to that region, so I should get this edge. And that's a more interesting change because as I draw this blue edge, this green edge is going to disappear. And so the green complex gets disconnected. And this is how we want to characterize what happens in the process. For example, a green cell goes through a circle of red cells. That changes the tunnels. So I can keep track of that. And then also when it backtracks again, low persistence. And so by looking at the topology of this thing, then I can read off which changes happened, how often, and to what persistence. And so this gives me a measurement and an observation. So I'm not in the business of explaining the process. I'm in the business of measuring it, and then hopefully the measurement is going to be useful in order to figure out why it happens and how it happens, and how to distinguish those things. All right, so if I run this algorithm and I compute a Voronoi diagram, just one color, I get some kind of space in 4 dimensions. So time goes upwards. So here's one point, has a trajectory. All the points have trajectories. And if I draw the cells around it, I get something which we call medusa. We invented the term medusa because it looks like a lot of hair. They're interestingly connected. And eventually, because there's cell segregation, they're all connected in one block. And the way this medusa is connected is characteristic of what happened in the process. There's a tunnel here. So this means a cell from the other type went through there for some time. So if I measure the connectivity, that's what characterizes things. All right, so this was a setup, and then here is a simulation to see whether this actually works. The simulation is done by, we have blue cells and red cells. And then we also have something that we call empty space, which is the outside. And then we say the blue cells like the red cells with a factor of 2. So positive numbers is liking. The red cells like each other a lot. The blue cells, okay. Both of them do not like empty space. Red cells are really disliking empty space, and blue cells a little bit. So this is like hydrophobic, hydrophilic perhaps. The reds are very hydrophobic. So you could imagine perhaps what happens when the cells have these kind of sticking factors that the reds try to separate away from the empty space. So maybe the blue cells will form a shell around them to protect them. In this setup, this is exactly what happens. So here is a random initial position, and then things move around. You see the cluster a little bit. Some cells lose the bulk and get separated. And then after some 70 steps or something, we have a sphere of blue cells around the red cell, around the population of red cells. And I wanted to go through a little bit to see whether we can see this in the topological measurements. And for this, I need to go back to the temporal interpretation. I myself find this interesting in general because I see myself walking through the world always seeing a level set at any time or through space-time. So I'm very interested in can I translate the little thing that I know in topology into some understanding of the world so that maybe sometimes I see something which I would have otherwise not seen. So I give you the opportunity to do the same exercise here. So we talk about temporal interpretation of homology groups. And there's a very simple classification. So in the ordinary diagram, a component I have to make a disclaimer here. In homology, we think of the zero homology as counting the components. It's a bad habit. We should really count the gaps between the components. It's like a tree with n nodes has n-1 edges. There are n-1 gaps. And this is much more natural in many circumstances, including this one. So let me think ordinary zero dimensions counts gaps. So here there's a gap opening up, and then the gap disappears because the cells merge. This is some kind of consolidation. In the one-dimensional ordinary diagram, I also see loops that form, and the loops fill up. Okay. So in the ordinary diagram, I see all the topology that aggregates. Somehow it forms at some point, and then it disappears by pushing things together, merging. In the horizontal diagram, which is the second triangle, if you remember, of my four triangle division of the big triangle. In the horizontal diagram, it's the same starting, but then it hesitates and it reverts to the original. So a gap forms, but then a gap disappears because one of the components dies. Or a ring forms, or a loop, and then somehow it doesn't fill up, instead it breaks up again. Hesitant aggregation. On the vertical side is hesitant disaggregation, because I have a block at the beginning, at the bottom. It breaks up by cutting it, and then somehow it changes mind and it merges again. It doesn't really break up. This is a vertical cycle, because we never see this one-dimensional cycle. We only see it in 4 dimensions, in space-time, but not in real space. And similarly, a one-dimensional ring it forms by puncturing and then disappears by filling up. And this forms a 2-dimensional cycle in 4 dimensions. I didn't say this right. My drawings are all for 2 and 3 dimensions. So I have 2-dimensional space and 1-dimensional time. All the computations are for 3 and 4 dimensions. So there's one extra case. So I think I misrepresented it a few times. In any case, the stuff in the relative diagram is disintegration or disaggregation. Things split up and they separate out. Okay, so here is how we read our diagram now. Separating ordinary, horizontal, vertical, relative. Dimensions 0, 1, 2, 3. The gaps are recorded in the 0-dimensional first 2 triangles and the 1-dimensional second 2 triangles. Just because in the second 2 triangles, that's the vertical stuff, we see only slices. So there we record the 1-dimensional thing, but we see slices, so they become 0-dimensional. So here we see the gaps. The tunnels we can read with the same step, and the voids we can read with the same step. Okay, so this is to translate the topology into the temporal interpretation. And then the words palatable for biologists is that things close and fill, break up, puncture. Left is aggregation, the right is disaggregation. In the middle is hesitant aggregation and disaggregation. And now I want to spend a few minutes on the results before we're done. So we measured this simulation. Here are the numbers. The blue numbers are the number of cycles of whatever dimension and class in the persistence diagrams there are. The real numbers with 2 digits are the accumulated persistence. So persistence, remember, is how long this interval was, or for a point, how far away it is from the baseline or from the vertical axis. And when we see, I want to focus a little bit. I want to focus on the I think I want to focus on these 61 points here. So you would expect to see a big void in the blue population. Oh yeah, I should also have said that above is the red diagram, the red medusa, blue medusa, and this is something else I explain in a second. But I want to focus on the blue medusa. So the blue medusa has a big void because it's around the red cells. But we don't see that. Namely, I show you the diagram. There is no high persistent 2-dimensional point. This is the 2-dimensional diagram. But what we do have in the horizontal diagram, we have this entire population of points here. And they come from the fact that the blue cells surround red cells, open up, they go back and forth all the time. So we don't see a single long interval where the blue cells form a shell. They add up to a lot of persistence, though, 0.76. I still would like to argue that there is actually one shell. It's just not perfect, and therefore my algorithm was not able to find it right away. For that, we use this last row. This is the inclusion of the blue medusa in the blue Voronoi medusa. To tell you the difference, the blue medusa is a union of spherical balls of a fixed radius, which approximates the cell size, sweeping out a medusa in 4 dimensions. So it has a certain thickness. If instead of actually using balls, but I take the entire Voronoi cells, then the medusa gets larger, so I have an inclusion. If I have an inclusion, this fills up all the gaps of imperfections that don't allow me to see it, the shell. So remember, we had 61 2 cycles, all of them short-lived. Together, they live for a long time. After this inclusion, we only have 1 2-cycle left. And this 2-cycle also has a large persistence. So this says if we use a more robust way of measuring the homology, in this case, the 2 cycles, we do, in fact, see the shell. And let's see where is this point? It's right here. Born early, die late. Yeah, that's the point. It's far away from the vertical axis, so that's its persistence. It's 0.95. It's normalized to 1, so it's almost the whole thing. And then you have a few other things. All right, and with this, I wanted to close my talk. Thank you very much.